Hello to all of you. So now we're going to discuss chapter number 10. Okay. So in chapter number 10, we are going to talk about DNA replication. So the first thing to think about is why the DNA has to replicate. So the DNA replicates because the cells in our body, they are dividing. Cells are dividing because we are born as a baby, then we go through different stages in our life. So we are born as a small baby, then we go through different stages like infant, toddler, adolescent, then we eventually become adult. So we pass through various stages of growth and development, and we are growing and developing because of cell division. Our body has some of the cells which the body is losing, new cells are forming, there is a wound healing, there are various things going on in the body, and for which our cells they need to divide. And for the cell division, it's very important that cell division takes by a process by cell division called mitosis. So in this process, so all the components of the cell they need to be duplicated and then they divide into half. So one of the very important component of the cell is the DNA, because DNA is having all the information. So this DNA needs to make its copy and then it is divided. So how actually DNA divides, how does DNA, duplicates or replicates? That's what we are going to study in this chapter. So in this chapter, we are going to focus on the DNA replication, and the next two chapters we are going to focus on the protein expression, which involves two steps: transcription and translation. Those I have already discussed in the previous lectures also, but now we are going to discuss it in more detail. So this is also called central dogma. This is the way how the information flows from DNA to RNA, from RNA to proteins. So DNA needs to be replicated for cell division, and DNA, as I've said, it's basically a knowledge, it's a information. It does not do anything by itself. Basically, the information in DNA is converted into proteins by the process of protein expression or gene expression, which goes through two different stages: transcription and translation. So in the transcription, the information which is there in DNA is converted into RNA, and the information which is there in the RNA, it is converted into proteins by the process of translation. So this is called central dogma, or this whole process is the way how the information flows in the cell. So in chapter number 10, we are going to focus on replication, and chapter number 11, we are going to focus on transcription, and on chapter number 12, we are going to focus on the translation. So basically, a replication, so it's, as you know, DNA is double stranded, there are two strands. So the mode of replication is also called semi-conservative mode. It's called semi-conservative because in this process of replication, like this is the DNA molecule which has two strands. The two strands, first of all, they separate. So it's shown here. This is two red strands. First of all, these two red strands will separate, and they both will act as the parent strand or the template strands, and whatever the sequence is there on the parent strands or the template strands, on the basis of that information, the new daughter strands are made, which are shown here in blue. So basically, the process of replication is the two strands, first of all, they separate, and both strands act as parent strand or the template strand, and whatever is the information on the parent strand, that information is used, and on the basis of that information, a new strand, a new daughter strand is made. So from one molecule of DNA, when two molecules of DNA they are formed, in each new molecule of DNA, one strand is old and one strand is new. That is why it's called a semi-conservative mode of replication. So basically, going more into the details of the process of replication. So replication process starts like this is your double-stranded DNA. So it starts from one place or one side, that is called the origin of replication. So DNA double helix unwinds at a specific point. So the two strands they will separate at one specific point, and that point is called the origin of replication. And then so the two strands separate, and then the new strands will be formed. They can be formed in one direction or in the both direction, and that results in the formation of a region which we call replication forks. So replication forks are basically Y-shaped regions. When they are the regions where the new polypeptide chains are being formed. So usually prokaryotic organisms like bacteria, they have only one origin of replication because prokaryotic organisms they have only one single chromosome. So they have usually one origin of replication, which means they have only one point where the two strands they separate. But in eukaryotes, because they have very, very long chromosomes, they have multiple origins of replications. So in the prokaryotes, there is only one origin of replication, and when the new DNA strands are being formed, they form a region which looks like a bubble, because the two strands separate and the new strands are being made on the basis of the sequence which is there in the template. It gives an appearance of a bubble. So in prokaryotes, there is only one origin of replication and one bubble. In eukaryotes, there are several origins of replication and several bubbles. So this is prokaryotic DNA chromosome. So you can see this is the origin of replication. There is one point from where the two strands they're going to separate. So you can see these regions and these Y-shaped regions we call replication forks, and this is one replication bubble. And in the case of eukaryotic DNA, because that's a very, very long DNA, there are multiple origins of replication. So there's one origin of replication here where the two strands have separated, second is here, third is here. So there are multiple origins of replications, so there are multiple replication bubbles. So you can see there's a one here, and one here. So the very, very important enzyme which is involved in DNA replication is called DNA polymerase. So something very, very important I would like to tell you about DNA polymerase is that this enzyme works in the direction of 5' to 3'. I hope you remember from the last lecture what 5' to 3' means. So if you remember, the two strands of DNA, they're antiparallel in nature. So if one strand is 5' to 3', the other strand is 3' to 5'. So on the 5' there are phosphate groups, and on the 3' there are hydroxyl groups. So the important thing to know about this DNA polymerase enzyme which is involved in DNA replication is that it always adds nucleotides. It always works in the direction of 5' to 3'. So it adds new nucleotides at the 3' end. So it always requires, so a new nucleotide will be added. So if this is the chain, this is a growing nucleotide chain, the DNA polymerase 3 is going to add a nucleotide at this position. This is the 3' of this nucleotide, and here is the hydroxyl group. So DNA polymerase is always looking for free hydroxyl group at the 3'. So it works in 5' to 3' direction. So please note down this number 53 because we are going to talk about all the processes, transcription, translation, they all work in 5' to 3' direction. So this is a new nucleotide which is being attached. So it's a function of DNA polymerase to add a new nucleotide. It always adds a new nucleotide at the 3' end. And the new nucleotide which is coming has 3 phosphate groups. So when the nucleotide is attached, the 2 phosphates are removed. This is called pyrophosphate. And this nucleotide, which is having only 1 phosphate, is going to be added to the growing chain of the nucleotides. So this is some, and also DNA polymerase, important thing to know about DNA polymerase 3 is that it cannot work on its own. It needs the help of a RNA primer. And what is the RNA primer? RNA primer is a short stretch of RNA. It's a short stretch of RNA which is made by another enzyme called primase, and then this DNA polymerase 3 is going to look for the 3' end, free 3' end of that RNA primer, and it's going to add nucleotides to that end. So DNA polymerase cannot initiate, but it can elongate. So the as you know, the 2 strands of DNA, they are antiparallel in nature. One of the strand can be made continuously, because remember that I just told you DNA polymerase works in 5' to 3' direction. So the strand which is 3' to 5', so the strand which is 3' to 5', that when that strand acts as the parent strand, the complementary strand, which is the daughter strand, which is going to be made on the basis of this strand, can be made in 5' to 3' direction by DNA polymerase. So this strand which can be made in 5' to 3' direction, that strand is made continuously, and that strand is called the leading strand. But the opposite strand, because of the antiparallel nature of the 2 strands, if one strand is 3' to 5', the opposite strand is going to be 5' to 3'. So DNA polymerase cannot make a daughter strand which is complementary to this strand, because DNA polymerase can add nucleotides only in the 5' to 3' direction. So the opposite strand is made in a different way. The opposite strand is made discontinuously. It is made in fragments, and those fragments are called Okazaki fragments. So this strand is called the lagging strand. So let's see here. So you can see here, this is a strand which is 3', and if this is 3', this side is the 5'. So DNA polymerase can make this strand going from 5' to 3'. That's fine, because that's its nature. DNA polymerase can add nucleotides from 5' to 3'. So this is the leading strand, and this strand can be made continuously by DNA polymerase. Now if you look at the opposite strand, this strand is 5' and this side is the 3'. So which means that this end, which will be here on the opposite strand, is going to be 3', right? So the DNA polymerase cannot make this opposite strand, this opposite daughter strand, in this direction. It cannot make it in this direction, because DNA polymerase makes the daughter strand, which is complementary to this strand, in fragments like this. Okay, so this, I'm not going to go into the details of this process, because the lagging strand, how it is made, it involves 2-3 enzymes. So just remember that this strand, it is made in fragments, and these fragments are called Okazaki fragments, and this strand is called lagging strand. So DNA polymerase makes this strand in this direction, because it cannot make in this direction. So this is the way how DNA polymerase works. So now let's go into more details of the enzymes in prokaryotic organisms, and then we'll talk about the eukaryotic organisms. So in E. coli, which is a bacteria, which is a prokaryotic organisms, there are at least 5 DNA polymerases, 3 of which are extensively studied. So polymerase 1 consists of a single polypeptide chain, and polymerase 2 and 3, these are multi-subunit proteins. So these are some of the characteristics. You can go over this table and see these are 3 different DNA polymerases in E. coli, and these are some of its characteristics given. But I would like to tell you something important which you should know, that for all these 3 enzymes is the polymerization 5' to 3'. All of these 3 can do the polymerization in this direction. Now what is exonuclease activity? Exonuclease activity means these enzymes, they do not only have polymerization ability, these enzymes also have an ability to remove the nucleotides. So that is what is a nuclease activity. So they also have, so these enzymes, they can remove the nucleotides in 5' to 3' direction, or 3' to 5'. So these enzymes, they can polymerize as well as they can also act as nucleases. So that's all the information given here. As I just said, DNA polymerase, it basically works in 5' to 3' direction, and the most important thing to know about this enzyme is it cannot initiate the process of polymerization on its own. What it requires a primer, and this primer is a short stretch of RNA, and this is made with the help of the enzyme called primase. So these are some of the requirements which are written here for the replication process. So for the replication process, these nucleotides are also required, because the daughter strand which is made, it is complementary to parent strand. And so basically this is how the replication takes place. It's carried out by replisomes. What is a replisome? It's a complex of DNA polymerase, RNA primer, primase, and helicase. Now what is a helicase? Helicase is that enzyme which helps in the separation of the 2 strands. So when the process of replication starts, the 2 strands of DNA, they are wound around each other. These 2 strands, first of all, they need to separate, and this process is called unwinding of the 2 strands of DNA, and that takes place with the help of the enzyme called helicase. Now what is DNA gyrase? DNA gyrase is an enzyme which basically helps in because when the helicase is opening up the 2 strands of DNA, because DNA is wrapped around each other, it can actually cause the 2 strands of DNA to be very tightly wound around each other as the helicase is opening the 2 strands of DNA. So the function of topoisomerase 2 or gyrase, DNA gyrase, is to relax this supercoiling of the DNA, because helicase is opening, and the DNA which is ahead of helicase, it can really become very tightly supercoiled. So that's a function of this gyrase. It basically relaxes these supercoils. Helicase, as I said, what helicase is, So these are all proteins which are involved in the process of replication. So it's important to know what are single strand binding proteins. So the function of these single strand binding proteins is to bind to the DNA. As helicase opens the two strands, these two strands can come back and they can wind around each other again. That's a possibility. But we have these proteins called single strand binding proteins. The function of these proteins is to, when the two strands they have separated, now they have become single stranded DNA. These proteins they go and bind, these proteins they go and bind on these single strands, and they do not let the two strands of DNA to come back again. So that's a function of the single strand binding proteins. We talked about what primase is. I have told you what primase is. So this is basically showing you how DNA replication takes place. So this is our DNA. So this is helicase. Okay, the function of helicase is to open the two strands of DNA. This is DNA gyrase, which will help in removing the supercoils, which will be formed in this region. And you can see this is the leading strand, and it is being made continuously. This is the lagging strand, which is made discontinuously, and basically shows all the different proteins which are involved. The single strand binding proteins, helicase, then in this direction primer is made. These are lagging strands. So this basically shows all the proteins which are involved, and I just showed you that. Okay. Yeah, this basically explains, and yeah, something important to know, the RNA primer, as I said, DNA polymerase cannot initiate the process of replication. It takes the help of this RNA primer, and this RNA primer is later removed, and that RNA primer that RNA primer is replaced by the deoxynucleotides. And as the lagging strand is made in fragments, and those fragments are called Okazaki fragments, and those fragments they are joined to each other by the enzyme called DNA ligase. This is the summary of the DNA replication which takes place in prokaryotic cells. You can go over it. Now we are going to talk about the DNA replication which takes place in eukaryotic cells. Now in the eukaryotic cells, it's a little more complicated. It's little because it involves some of the more important proteins. So in the eukaryotic cells, they go through a cell cycle. So basically, the cells uh, go through a long phase. We call it an interphase. In this phase, or it's also called a preparatory phase. So in this long phase, cells are actually preparing for the upcoming division. And in this phase, as I said, that before the cell can divide, there's a preparatory phase. In that phase, cell actually is doing the preparation. It is duplicating everything. So this phase, interphase or preparatory phase, goes through these phases. There's a phase called G1, S, and G2. In G1, it's basically duplicating all its organelles. In S, it's duplicating its DNA, and in G2, it's doing the further preparations. So the DNA replication actually takes place here, and followed by interphase is mitotic phase, and the mitotic phase basically cell divides. So this is the place where the DNA replication actually takes place. So basically, in the eukaryotic replication, it involves few more proteins like origin recognition complex. So it's a complex of some proteins. They basically go and bind to the origin of replication, and then there are some another proteins which are involved. That's a replication activator protein, replication licensing protein. So these are all proteins which are involved in the eukaryotic replication, and they form a pre-replication complex. And then in this process also there are some more proteins involved. They're called cyclins and cyclin dependent kinases. So this is a cyclins and cyclin dependent kinases. They are kind of a complex. They are also proteins. So cyclins are proteins whose concentration keeps changing in the cell cycle. So that's why they're called cyclins. And cyclin dependent kinases are enzymes. Kinases are enzymes which can transfer the phosphate groups, and they get activated only when they have their partner with them, the cyclin. So this cyclin dependent kinase will function as an enzyme, and as an enzyme it transfers the phosphate groups. It will function only when it is conjugated with this cyclin. So so the replication takes place in the S phase. So this is basically showing you uh, how the different proteins are involved in the process of DNA replication in eukaryotes. So this is, you can see, this is early G1, which is before S phase. So you can see this is your DNA, and this is the ORC complex proteins, the which we just discussed here, the origin recognition complex. So they all come and bind here. Then they're followed by these proteins, the replication activator proteins, and then they're followed by all these other proteins, and they form a pre-replication complex here. And then, so this is blue thing, this is DNA, and these are all proteins. They're getting assembled here and forming a pre-replication complex. And then there's the involvement of these kinases, cyclin and cyclin dependent kinases, and they are the ones which are going to phosphorylate these proteins. Okay, they are the ones which are going to phosphorylate all these proteins, and then these proteins, they get phosphorylated, and then the DNA replication takes place. So basically this is something different in the eukaryotic cells. This basically shows the different proteins which are involved in the replication of DNA in the eukaryote. So these all proteins are involved, and they make the DNA replicate in the S phase. So all these proteins, they get assembled, and then with the help of these kinases, they get phosphorylated, and then they make the DNA replicate in S phase. And this basically shows some of the polymerases which are found in the eukaryotes, and 5 out of them are extensively studied. So they basically involve alpha, then delta, then epsilon, beta, and gamma. So these are different DNA polymerases, and these are some of their characteristics. So these are the 5 eukaryotic DNA polymerases, and is the most important functions of them is polymerase alpha is involved in making primers, then delta is involved in which is the principal DNA polymerase in eukaryotes. Epsilon is involved in the making of a leading strand. Beta is basically involved in repair, and gamma is involved in the DNA replication in the mitochondria. And these are some of the differences in the DNA replication processes in the prokaryotes and the eukaryotes. And this is basically showing the full replication machinery in the eukaryotes. So it's basically involving, so this is the leading strand, this is the this side is the lagging strand. So this is the formation of a RNA primer here. This is a helicase, then this is the topoisomerase. These are all proteins which are involved, RFC, polymerase delta, which is the main polymerase. So this is the whole replication machinery which is involved in the DNA replication in the eukaryotes. Okay. So that's all about this chapter. If you have a question, please let me know.